You know, inside every single one of your cells, there's a blueprint. It's more complex, more elegant than anything humans have ever designed. It's the code of life itself. And today we're going to pull back the curtain and take a look at the incredible engine room of biology, molecular genetics. So here's the game plan. We'll start with the master document, DNA, and see how it's all stored. Then we'll look at how cells make perfect copies of it. We're going to follow the message as it gets transcribed into RNA, then watch the assembly line that translates that message into a protein. And finally, we'll open up the amazing toolkit that scientists now use to read and even rewrite this code. All right, so let's dive right in with the master document itself, the blueprint for every living thing on this planet, DNA. So what exactly is DNA's job? Well, it really has two absolutely mission-critical roles. First, it's a master of copying itself. It's like a biological photocopier that makes sure every new cell gets a perfect set of instructions. And second, it holds the genes. These are the unique individual recipes for building every single protein your body needs to function. But where do you keep such a vital blueprint? It turns out not all cells use the same filing system. There's a really fundamental split between simple cells called prokaryotes, like bacteria, and the much more complex cells that make you, the eukaryotes. And you can really see that difference here. On the left, in a prokaryote, the DNA is just a single circular chromosome kind of floating around inside the cell. You'll also see these little extra loops of DNA called plasmids, which can carry some bonus genes. But then look at us eukaryotes on the right. We are masters of organization. Our DNA is split into multiple, linear chromosomes, and it's all packed away securely inside a nucleus. To make it all fit, it's wound incredibly tightly around these proteins called histones. It's like trying to pack miles of thread onto a bunch of tiny little spools. So when we talk about a gene, we're not talking about that whole massive chromosome. A gene is just one specific section of that super long, coiled-up DNA molecule, and each one has the exact instructions for one single protein. The best way to think about it is this. A chromosome is like a giant cookbook, and each gene is just a single recipe inside it. Okay, so if every new cell needs its own copy of that cookbook, how does the cell go about printing one? That brings us to the molecular copy machine, DNA replication. The process is actually super elegant, and it's called the semi-conservative model. Semi for half, and conservative for save. Basically, the DNA double helix gets unzipped, and each of the two original strands is used as a template to build a new matching partner. What you end up with are two perfect DNA copies, and each one has one of the old strands and one brand new strand. It's just a beautiful system for making sure the copy is flawless. Now, this whole operation requires an all-star team of enzymes. First up is helicase. That's the unzipper. It unwinds the double helix. Then primase comes in and lays down a little primer, which basically says, hey, start copying right here. The star of the show, though, is DNA polymerase. That's the master builder. It reads the template and adds all the new DNA bases. And finally, you've got ligase, which is like molecular glue, coming in to seal up any gaps and make sure the new strand is perfect. Now, here's a fascinating little quirk in the process. That master builder, DNA polymerase, it's like a machine on a one-way track. It can only build in one direction. For one of the new DNA strands, the leading strand, this works out perfectly. It just gets built in one long continuous piece. But the other strand is facing the wrong way. So the lagging strand has to be built backwards in these short little chunks that then get stitched together later. Pretty clever, right? Okay, so we've made a perfect copy of the DNA blueprint. Now that blueprint is kept super safe in the nucleus. Think of it like a protected library. But the factories that build the proteins, the ribosomes, they're out in the main part of the cell. So how do you get the recipe from the library to the kitchen without damaging the original priceless book? Simple. You send a messenger. And that messenger is RNA. It's a very close cousin of DNA, but there are three key differences. It uses a slightly different sugar, it's usually single-stranded instead of double, and, this is a big one, it swaps out the DNA base thymine for a different base called uracil. So anywhere you'd see a T in DNA, RNA has a U instead. And there isn't just one type of RNA, it's actually a specialized team. You've got messenger RNA, or mRNA. That's the actual disposable copy of the gene's recipe. Then there's ribosomal RNA, or rRNA, which is a building block of the ribosome factory itself. And finally, you have transfer RNA, or tRNA, which acts like a delivery truck, bringing the raw materials, the amino acids, to the assembly line. Now, before our messenger RNA is ready to go, it needs a little bit of editing. The first draft has these sections called introns, which are kind of like gibberish in the middle of the recipe. So a process called splicing cuts out all those introns and pastes the meaningful parts, the exons, back together. The cell also adds a protective cap and tail to make sure the message doesn't get damaged on its journey. It's pretty amazing. So, with our edited and protected message in hand, it's finally time to head over to the cellular assembly line and actually build something. This process is called translation. So how does this assembly line actually work? Well, the finished mRNA finds a ribosome and docks with it. The ribosome then starts reading the mRNA sequence in three-letter words called codons. For every single codon it reads, a specific tRNA molecule shows up, carrying the exact amino acid that matches that three-letter code. The ribosome then links these amino acids together, one after another, building a chain that will eventually fold up into a complex working protein. You know, this deep understanding of the entire process, from DNA to RNA to protein, has given us an incredible new power. We now have the ability not just to read the code of life, but in many ways, to actually edit it. So let's take a peek inside the geneticist's toolkit. This brings us to a revolutionary idea: recombinant DNA. This is really the heart of genetic engineering, creating DNA molecules in a lab that combine genes from different organisms. The classic example is taking the human gene for insulin and sticking it into one of those bacterial plasmids we talked about earlier. This turns those bacteria into tiny, living insulin factories. So how do you perform this molecular cut and paste? You use tools called restriction enzymes. You can think of them as tiny molecular scissors that are programmed to cut DNA only at a very specific sequence. So, by using the same enzyme on both the gene you want and the plasmid you're putting it into, you create perfectly matching sticky ends that let the new piece of DNA slot right in. But after all this cutting and pasting, how do you sort through all the tiny DNA fragments you've made? One of the most fundamental techniques is called gel electrophoresis. See, DNA has a negative charge, so if you put it in a gel and run an electric current through it, the DNA will start moving toward the positive end. The gel acts like a dense forest, so smaller fragments can zip through pretty fast, while the larger ones get tangled up and move a lot slower. This lets us sort all the DNA fragments by their size. And this seemingly simple technique has huge, real-world applications. It's the basis for DNA fingerprinting that forensic scientists use at crime scenes. It's used to establish family relationships, like in paternity tests. And on a much bigger scale, it helps scientists map out the tree of life by comparing the DNA of different species to see how closely they're related. So we've taken this journey all the way from the discovery of a simple double helix to developing a powerful toolkit that literally allows us to rewrite the source code of life itself. The power here has an almost unimaginable promise for curing diseases and solving huge global challenges. But it also hands us an enormous responsibility. Now that we can truly read and write the code of life, the biggest challenge we face is deciding what exactly we should be writing.